Good day. Today we have our second lecture, and topic today will be very difficult, but very necessary for your next practice. Okay, anatomical and morphological, histological and X-ray features, radiological features of the teeth jaw system in children at different ages. Let's start. What we need to know? We need to know that although understanding of the histology, physiology, and occlusal interaction of the dentition and surrounding tissues of the teeth, it's very necessary knowledge, yes, and this knowledge of the structure of teeth, for example, enamel, dentin, cementum, and pulp, and their relationship to each other and to the supporting structure is necessary, especially when we treat our teeth. The protective function of the tooth form is revealed by its impact or masticatory muscle activity, the supporting tissues, and of course the pulp. Teeth and supporting tissues, periods of occlusion in children. We have three types of period. It's first type of bite. First type is temporary bite, and here you can see three periods also. First, it's a period of formation, then period of stable temporary occlusion, and period of again late temporary bite. The second variable bite, two periods, has this variable bite, early and late. And the third is a constant bite. Development of temporary teeth. Of course, we have 5 steps here. First, it's bookmark and intramaxillary formation, then eruption of the teeth, formation of root and periodontium, stabilization, and resorption. Sorry, here we have some mistake. Resorption, guys, 5. The period of intramaxillary formation. Formation of dental plaque and dental buds, differentiation of dental germs, and histogenesis of tissues. Histogenesis of tissues. Next one, formation of the dental germs. We know that dental germs, we can see the development of them at 6-8 weeks of development in the area of future dental arches of the upper and lower jaw. Okay, here you can see that the first is gum epithelium, the second dental plate, here plate, guys, then mesenchyme tissues and dental kidney or dental germs. At 10 weeks of embryonic development in each epithelial kidney begins in grow mesenchyme tissues is formed dental epithelial, so enamel organ. In your tests, when you have time for preparation for a lesson, you will have the tests, and these tests you will have on practical lessons, and when you will prepare to license exam Krok 1. And this knowledge you need to write in your synopsis. The mesenchyme tissues which grows into the epithelial organ is called dental papilla. And dental bag, the mesenchyme surrounding the enamel organ. Here you can see the table, differentiation of the tooth rudiments, and a dental plate here, enamel organ, then fourth, it's internal cells of the enamel organ, internal, okay? Next one, outer cells of the enamel organ, okay? Layer of odontoblasts. Then dental papilla, the edge of enamel organ, and dental sac. And of course, the next table is also development of tooth tissues. Not very difficult. Here also outer cells and internal cells. You can also write in your synopsis. Okay, tooth structure. Enamel, dentin, then pulp. Okay, and it is this part, it's crown of the teeth, neck of the teeth, then radix or root, cementum tissue, it's our bone, and the gums. Enamel. So, enamel formation, that we called also emelogenesis, is accomplished by cells that we called emeloblasts. These cells originate from the embryonic germ layer known as ectoderm. Enamel covers the anatomic crown of the tooth and varies in thickness in different areas. And it is securely attached to the dentin by the dentino-enamel junction. It is thicker at the incisal and occlusal areas of the crown and becomes progressively thinner until it terminates at the cementum enamel junction. Then, chemically, enamel is a highly mineralized crystalline structure. Hydroxyapatite is the form of a crystalline lattice. It's the largest mineral constituent. Yes. So other minerals and trace elements are present in smaller amounts. The remaining constituent of tooth enamel include organic matrix proteins. It's 1–2%, and water from 4 to 12%. Structurally, enamel is composed of millions of enamel rods or prisms, rod sheaths, and cement in interrod substance. Enamel rods, which are the largest structural components, are formed linearly by successive apposition of enamel in discrete increments. So the resultant variation, you can see enamel prisms here, and a dentin enamel junction. The resulting variations in structure and mineralization are called incremental stria of ratios and may be considered growth rates that form during amelogenesis. The stria of ratios appear as concentric circles in horizontal section of a root. In vertical section, the stria are positioned transversely at the cusps and incisal areas in a symmetric pattern. Then, elevation between the growths are called pericumata. They are continuous around a tooth and usually layer parallel to the cementum enamel junction and each other. Rods vary its number from approximately 5 million for mandibular incisors to about 12 million for maxillary molar. Then, enamel structure. Of course, we know that enamel structure, also here Guntel Schrager stripes and ratius line. Okay, I tell you now. And enamel rod diameter near the dentinal borders is about 4 microns and about 8 microns near the surface. This diameter difference accommodates the larger outer surface of the enamel crown compared with the dentinal surface at the dentin enamel junction. Okay, next one. Ameloblasts degenerate upon completion of Naismith membrane. Naismith membrane. This membrane or a primary enamel cuticle, which covers the newly erupted tooth and is worn away by mastication and cleaning. The membrane is replaced by an organic deposit called the pellicle, which is precipitate of salivary proteins. Microorganisms may attach to the pellicle to form a biofilm or bacterial plaque, which, if exudogenic in nature, may become a precursor to dental diseases. Each enamel rods contain millions of small elongated apatite crystallites that vary in size and in shapes. So, although enamel is a hard and dense structure, it is permeable to certain ions and molecules. The route of passage may be through structural units, such as rods, sheets, enamel cracks, and other defects that are hypermineralized and rich in organic content. Then enamel is soluble when exposed to acidic condition, but the dissolution is not uniform. Solubility of enamel increases from the enamel surface to the dentin and enamel junction. When fluorine ions are present during enamel formation or are typically applied to the enamel surface, the solubility of surface enamel is decreased. Okay, enamel is the hardest substance and tissues in the human body. Hardness may vary over the external tooth surface according to the location. Although it decreases inward, with hardness lowest at the dentin enamel junction. Okay, next one. The dentin. The dentin, inorganic substance, we know that it's, for example, 17 or 72%. Organic substance and water, 28 and 30. And here, inorganic and organic substances, you can see the example. So, inorganic is calcium, calcium phosphate, calcium carbonate, calcium fluoride. Then organic, it's collagen, 90%, phosphoroproteins, glycoproteins. The structure of the dentin, of course, the main structure elements of dentin is collagen fibers and basic substance and dentinal tubules. We know that in dentin we have different layers. In particular is outer layer or cloak dentin or it's collagen fibers, yeah, or Corfu fibers. Have a radial location. Inlayer or it's dentin near the pulp, and collagen fibers, we call them Ebner fibers, have a tangential arrangement. And intermediate layer also we have in dentin. And dentin formation or dentinogenesis is accomplished by cells that we called odontoblasts. Odontoblasts are considered part of pulp and dentin tissues because their cell bodies are in the pulp cavity, but they long, slender cytoplasmic cell process, tomes fiber we call them, extend when from 100 to 200 microns into the tubules in the mineralized dentin. Because of these odontoblastic cell processes, dentin is considered a living tissue which their capability of reaction to physiologic and pathologic stimuli. Odontoblastic processes occasionally cross the dentin and enamel junction into enamel. These are termed enamel spindles when their ends are thickened. Then dentin forms the largest portion of the tooth structure, extending almost the full length of the teeth. Externally, dentin is covered by enamel on the anatomical crown and cementum on the anatomical root. Internally, dentin forms the wall of the pulp cavity, or we call pulp chamber and pulp canals. Dentin formation begins intermediately before enamel formation. Odontoblasts generate an extracellular collagen matrix as they begin to move away from adjacent ameloblasts. Mineralization of the collagen matrix facilitated by modification of the collagen matrix by various non-collagenous proteins gradually follow its secretion. The most recently formed layer of dentin is always on the pulpal surface. The dentinal tubules are small canals that remain from the process of dentinogenesis and extend through the entire lines of dentin from the pulp to the dentin and enamel junction. Each tubule contains the cytoplasmic cell process of the odontoblasts and is lined with a layer of peritubular dentin, which is much more mineralized than the surrounding intertubular dentin. Okay, the next one slide. Peritubular dentin, interglobal dentin. You can see the preparate, and it is peritubular dentin. Okay, and interglobal. Predentin, it's a dentinogenic area. It's a very thin layer, the dentin that are mineralized and located on the border with the pulp. And also a picture, a photo from the microscope. After the primary dentin is formed, dentin deposition continues at a reduced rate, even without obvious external stimuli, although the rate and amount of this physiological secondary dentin vary considerably among individuals. In secondary dentin, look please, primary, secondary, and tertiary. In secondary dentin, the tubules take a slightly different directional pattern in contrast to the primary dentin. The secondary dentin forms on all external aspects of the pulp cavity, but in the pulp chamber, in multi-rooted teeth, it turned to be thicker of the roof and flow then on the side walls. The walls of the dentinal tubules or peritubular dentin in the primary dentin gradually thicken through ongoing mineral deposition with age. The dentin therefore becomes harder, denser, and because tubular fluid flow becomes more restricted as lumen spaces become smaller, less sensitive. And the tertiary dentin, it's a dentin replacement or irregular dentin, which is formed during different pathological stimuli. For example, when patients have caries or some trauma and increased abrasion. And pulp. What we know about the pulp, that we have two parts, coronal and root pulp. And we need to know that pulp and dentin tissues are specialized connective tissues of mesodermal origin and formed the dental papilla of the tooth bud. And many investigators consider these two tissues as a single tissue. Many investigators consider this say us about the forms the pulp-dentin complex, which mineralized dentin constituting the mature and product of cells differentiation and maturation. So, dental pulp occupies the pulp cavity in the tooth and is is a unique specialized organ of the human body that serves for function. And histological structure, look please. First it's dentin, second predentin layer, then peripheral layer of the pulp, intermediate layer of the pulp, central layer of pulp, okay, and it is odontoblast process and the body of the odontoblast. And about the function I want to tell you, 4 functions: formative or developmental, then nutritive, sensory or protective function, and the last one, it is a defensive or reparative function. The formative function in the production of primary and secondary dentin by odontoblasts. The nutritive function supplied mineral ions, proteins, and their processes. The sensory function is provided by nerve fibers within the pulp that mediate the sensation of pain. Dentin nervous nociceptors are unique because various stimuli elicit only pain as a response. The pulp usually does not differentiate between heart, tone, pressure, or chemicals. The pulp is circumscribed by dentin and is lined peripherally by a cellular layer of odontoblast attachment to dentin. Anatomically, the pulp is divided into first coronal part of pulp, and this part located in the pulp chamber in the crown portion of the tooth, including the pulp horns that are located beneath the incisal ridges and cusp tips. And second one is radicular pulp that located in the pulp canals in the root portion of the teeth. The pulp contains nerves, arterioles, venules, capillaries, lymph channels, connective tissue cells, intercellular substance, odontoblasts, fibroblasts, macrophages, collagen, and fine fibers. The pulp is circumscribed peripherally by a specialized odontogenic area composed on the odontoblast, the cell-free zone, and the cell-rich zone. Okay, the cement, inorganic substance 68%, organic matter and water 32%. Also two types, cell-free and cellular, primary and secondary. The first covers the entire surface of the root, and the second covers the apical third of the root or bifurcation. Then, features of the structure of temporary teeth. Also, we know that the crowns of the teeth are low and wide. The color in some areas of the teeth are milky white or have such blue tingle. Then in the neck there is thickening and an enamel roller. Significant difference in molar roots. Then cell-free cement predominates and relatively large volume of the tooth cavity. Root canals wide, short, ending in several wide apical holes, and the periodontal jab is much wider. Enamel prisms shorter. Enamel contains more organic matter, their degree of mineralization, then hypermineralized areas of the enamel, and dental tubes are white. The pulp has more cellular elements but less focused their location. About eruption, it's a process of vertical movement of the tooth from the place of its tap and development inside the jaw to the appearance of a crown in the mouth. And eruption of the temporary teeth starts at 5-6 months of life and ends up to from 2 to 2.5 years. Dentition from 4 months to 2 years is called early. From 8 to 10 months and to 3.5 years we call late. Signs of physiological eruption, of course, it's timeliness, sequence, parity, and painless. And system Kent-Mondi-Palmer. Okay, here you can see the temporary teeth and permanent teeth, and the numerals of these teeth, horizontal layer, and vertical line. So vertical line, it's we have right and left side. Head wrap system. Also we have permanent teeth and temporary teeth. And it is special dental formulas that we write in the register card of the patient. The World Health Organization system, it's very useful in the register card, and it's this formula give us the also numerals of teeth. So we have 4 segments, first, second, 3, and 4. And then we have its permanent bite. And then we write, for example, 11, it's the first central incisors at the right. Okay, we see as a patient sit. So it's right part, left part, and right part. Upper jaw and low. And then we, for example, write 16. Yeah, 16 is the first permanent molar on the right part. And what about temporary? 5, 6, 7, and 8 segment. And also 51, 52, 53, its number of tooth. Then universal system. Here we write only 1, 2, 3, 4, 5, 6. We have only numbers from 1 to 17, to 16, and 32. Okay. If you have some question in practical lessons, we will speak about the dental formula more detailed. Universal anatomical system. Not very difficult system, but also useful. Constant bite and temporary bite. So letter is the name of the teeth. Incisors, canines, premolars, and molars. In the permanent bite we have a big letter, in the temporary small. These are some features in the structure of crowns and roots of teeth which allow you to determine whether they belong to one or another side, upper or lower jaw. Crown angle mark the angle formed by the medial surface and cutting edge of the front teeth, and sharper than the angle formed distal or lateral surface and cutting edge. So the same sign applies and to the masticatory group of teeth. Okay, distal surface and medial. And medial part of the vestibular side crowns more convex than distal. Apex deviates are in distal side relative longitudinal axis. Okay. Root and periodontal formation. Stage first. So now we speak about the steps of root formation. It's very necessary for your practice, because when we will have pulpitis or periodontitis and we work in the root canals and have endodontic treatment, it's very necessary knowledge. So stage 1, incomplete growth of the root in length. So for example, when teeth has eruption, it is first molar. Okay, first molar, for example, the start of eruption from 6 to 7 years, and we give for root formation 2 or 2.5 years. And in this period we have the different stages of formation. And first stage is it's a complete growth of the root in length. So we know that the root needs to be longer. The root is short, its walls are projected in the form of 2 parallel light strips, which gradually synex inspected in the direction of the germinal zone, forming a bell. The second stage, unformed top, root of normal length, the walls are parallel, gradually thinning towards the top. The periodontal fissure has a uniform width, merges with the growth though. Look please. So unformed top. Then open apical opening. We have apex, open apex. So root walls approaching in the apical part but not completely closed. The root canal is wide, the periodontal jab extended to the top. The fourth stage, unformed periodontum, deformation of the periodontal fissure. Okay, it's periodontum tissues, and we can see that it is unformed now. And the 5, it's fully formed root and periodontum. Periodontum fissure, cementum, periodontal fibers. Okay, and the stage of root and periodontal formation lasts from 1.5 to 2 years. It's for incisors, and 2.5 years for canines and for molars. Stabilization. The period is a functional full-fledged temporal bite, lasts from 2 to 2.5 years. Resorption period. From the age of 5 to 6 years, a temporary bite is replaced by a permanent one. Resorption of the roots of temporary teeth begins in the area where the nearest rudiment is located, permanent tooth, and it's also necessarily knowledge for we treat these teeth or we need to do extraction. Types of physiological resorption of roots temporary teeth, you can see 3 types. First types, it's uniform resorption, 3 roots. Then uneven resorption, only one root. And the third type of resorption in the area of bifurcation. Terms of development of temporary teeth. This table you need to write in your synopsis because in practical lesson we will speak about it. And you need to know the terms. Here you can see the table, the first radiological signs of tooth crown mineralization, then a term of eruption, end of root formation, and beginning of tooth resorption. It's for it's for temporary teeth, sorry. Stage of development of permanent, also bookmark and intramaxillary formation, eruption, formation, and stabilization. Here we don't have the stage resorption of the teeth. Then at 24-25 weeks of pregnancy, we start formation of first permanent molar. At 8 months of fetal development in a bookmark, permanent incisors and canines, and at the birth of the child, 16 permanent teeth. The beginning of the first premolar, late on the two year of the child's life. Then another premolar and beginning of another molar. Here you can see years. In terms of development, the permanent teeth, also all teeth you can see, but beginning mineralization, end of crown formation, cutting, and the root growths and formation. It is orthopantomogram of the child, and here we have variable period of bite, and we can see, for example, the first molar, permanent. We can see the germs of the permanent teeth. We can see the length of the stage, the length, the growths of the teeth in the lens, and resorption of the teeth of the roots. And the orthopantomogram of the child in permanent period bite. We have germs of the 8 teeth, feeling, and that's all. The development of temporary and permanent teeth occurs in the same way but at different times. In particular, when the temporary teeth are in a late stage of development, there are the rudiments of permanent teeth which are in the early stages of development. And literature that we have in our department, but not all, unfortunately, not all of this literature book we have in English. So if we write in the dean's office, I think they work with this problem, and I think that you will have books. Thank you for your attention, and please guys, if you have some question, of course you can write us under the video, and we will speak on our practical lessons more detail, because today we have very big topic and we can't to speak about of all of these tissues, of enamel tissues, pulp, dentin, very detailed. So it's a short knowledge lesson today. Thank you for your attention. Goodbye. Have a nice day.